Hello and welcome to another lecture of the course Aquaculture. In continuation to my previous lecture, now I am going to discuss about second part of the culture methods of Penaeus monodon, the tiger prawn. Uh, so far in the previous lecture, we have discussed about pond culture, then we also discussed about extensive systems, semi-intensive and intensive system, then we discussed about um, RAS, recirculatory aquaculture system. Now coming to another very promising type of culture system, that is biofloc system. We can utilize biofloc system in case of fishes, we can utilize biofloc system in case of prawns also. In fact, what is there in biofloc system? Uh, this is very emerging technology, we can say, where we can have the advantage of microbes. And somehow what we have to do in this, we have carbon and nitrogen ratios need to be balanced in biofloc systems in order to promote the growth of heterotrophic bacteria. What they do, heterotrophic bacteria, they use ammonia in the system, culture system, to grow and release nitrogen gas from the system. So that is one of the biggest advantages, that we are not purifying the water outside that system, but within the system. The growth of heterotrophic bacteria, which will directly consume the toxic ammonia, in fact, they will utilize from the culture system. And what they do, they subsequently produce a cellular protein which can form food of the culturable organisms. So, for having a better biofloc system to lead to the production of cellular protein and utilization of ammonia, we need to maintain carbon to nitrogen ratio of 10 to 20. That is usually recommended. And this is the reason that this biofloc system, which is microbiota based, that we have already discussed, is considered environment friendly, and it improves the water quality and disease resistance of the culturable organisms. It can be done in indoor cemented tanks of measuring varying sizes that you can have according to the number of individuals that you wish to culture. You can have you can manipulate, in fact, the tank size and the number of individuals that you wish to have. But it should be taken care that this tank should be painted or do something else to provide a smooth inner surface, or you can use another material. If you are not using any material which needs to be painted, then again this is fine. The tank should be then rinsed with disinfected with seawater. Rows of PVC pipes, these plastic pipes, PVC pipes of 1 inch, perforated L-shaped pipes laid on the tank bottom to serve as lift aeration system. In fact, you just perforate them, have little small perforations at the bottom, put it there, and then when air causes, aeration of the system becomes very easy. Blowers may be used for aeration purpose, using this throughout the culture of ensuring better bioflocculation to maintain the solids in suspension and to ensure that dissolved oxygen remained at saturation or near saturation levels. It's not good to say saturation level, but near saturation levels, you can say. In other words, you can say that dissolved oxygen content should be at a higher level. So that is always better. Bioflocculation, in fact, refers to a natural process that utilizes or you can say that uses microorganisms or their polymers to help with flocculation. And flocculation, what is flocculation? That is a process that causes small particles in a liquid or a solution to clump together, getting flocked into larger clusters known as flocks. This process can happen naturally or by adding a clarifying agent or flocculant to the liquid. So to this system, we are not adding those, but we are taking help of microbes. So microorganisms, like it's quite effective and environmental friendly way to harvest microalgae or treat wastewater Microorganisms like bacteria, algae, and fungi secrete extracellular polymers during their growth phase or cell lysis. These polymers are made up of polysaccharides, protein polymers, glycoproteins, and lipids. So, the functional groups of in these polymers in fact provide binding sites for pollutants. That is the main concept, which helps with flocculation. So, that is how this is carried out, the concept behind biofloc, how biofloc works. So, this way you must have got the idea that how it works. Before starting the culture, therefore, we need to add something which can initiate biofloc production or which can cause biofloc production. What are different ingredients which are used? 0.6 gram urea, 10 gram soybean, 2 gram rice bran, 0.5 ml molasses, 0.2 gram NH4 PO4 per liter of seawater should be added in a 400 LFRP fiber reinforced plastic tank for biofloc production. That is the finding of some of the researchers, but again go with the range and always go with the literature which can be giving more precise data for your region, and little bit manipulation will be required. Not all ingredients you will have to worry about, they will remain the same, but with the dosage a little flexibility is always there. So, this is the system, in fact, the diagram, the layout of how these biofloc systems, in fact, the experimental setup of the biofloc system looks like. If you know the concept, if you have the concept, then you can have, again, I should say, a little bit modification based on the same layout or same concept. Healthy and disease-free Penaeus monodon PL post-larvae of about 0.18 grams should be stocked in biofloc tanks. In fact, for any culture system, we are using post larvae only. They are quite hardy and they give good growth rate. Stocking density here we can have quite good stocking density, 400 to 500 PL per cubic meter of water. Feeding 3 to 4 times a day. That is regarding feeding level only. You can feed ration, you will have to adjust. Then 2 gram per cubic meter of commercial probiotics can be added to each tank at fortnightly intervals. Probiotics always provide, boost the growth rate. You can use some kind of immunostimulants also. So, that can be done. 10% of water exchange every 2 weeks. See, very little water exchange. 10%, only 10% of water exchange every 2 weeks by removing the sludge from the culture water. Here sludge is, separated. So, ultimately, you have to remove that. Water monitoring should be done regularly. That is something, we are culturing aquatic organisms. So, ultimately, their ambience, their environment need to be taken care of if we want them to perform in a better way. So, water monitoring should be done regularly, continuously. You should be very alert for monitoring. What happens? Sometimes we become, we take it for granted and we focus only on feeding. Go on feeding and feeding and feeding and don't care about water quality. Ultimately, we are going to get good result. No, you will not get. Even in semi-intensive aquaculture, if you are not having your eye on the fluctuating water quality, if you have added too much of food, then that become a problem. Here, you are doing everything in a very scientific manner. Here, you will have to be more alert. Survival rate, that was with some of the experiments, and growth rate, this much growth rate was obtained. That was quite good. What are advantages of biofloc system? Reduced water treatment expenses. In case of RAS, we were having more water treatment steps. Here we are not having that. We are utilizing microbes. Microbes are also providing food to the culturable organisms, and that means they ultimately reducing your feed cost, and they are making the efficient utilization of feed which you supply them. Lower feed cost, reduced pathogen risk because you are doing it very efficiently in an organic manner, enhanced growth performance and survival. What are limitations? Continuous aeration is must. So that will of course add to your cost. Need for skilled personnel for operation and maintaining the system. A little bit of skill and expertise will be needed. Not affordable for small scale shrimp farmers due to technological advancement, but it should not be said like not affordable. We may, we may afford it if we have, access to different kind of government schemes. Government is now having a variety of schemes that make these kind of systems, advanced systems, affordable to our Indian farmers. And we should, visit different agencies, the web page of different agencies that I have talked about in my previous lectures, so for getting such kind of advantages. So in the culture of method of prawn, we have so far talked about the culture systems. Now, without the seed, I have kept it, in the later slides, because the supply of seed, the overall breeding technology of the organism which you are culturing, is the key in order to decide the success of the culture practice. So, how we undertake induced breeding? The first step is to procure brood stock, to get brood stock. Where from to get brood stock of Penaeus monodon? You can get it from wild or from ponds. You can raise it from ponds, but wild caught brood stock are always in very high demand. So for wild brood stock, we are dependent on tides, and tide stationary gears are used, or sometimes by trawlers they are caught, or fishing boats. There are some divers. So you can get by any means the wild brood stock. And from wild brood stock from brackish water area should be avoided, as they give poor hatching rates. But wild brood stock from offshore water area should be preferred. They always give better breeding performance compared to near shore regions. Wild brood stock give a faster turnover and produce in 4 to 8 weeks the same number of larvae that pond brood stock yield in 8 to 12 weeks. So it's always better to go for wild brood stock if you can get, or you can work out if there is some technology which comes up and provides equally better performing pond raised brood stock, then that can also be undertaken. Transportation of prawns is done if you have to transport it from one place to the other. That can be done in seawater provided with proper aeration, and the temperature is kept little lower. So sometimes ice is also used to lower the temperature of the water. So depending on the conditions, that can be done, that how much temperature is there. So that way you can take care. The temperature should not be too high. High temperatures we avoid. And stocking in 1 ton capacity tank during traveling. 1 hour or less travel, you can stock 400 prawns if the water tank is of 1 ton capacity. Then you can have 400 prawns if you are traveling only for 1 hour. If for 4 to 5 hours, then you will have to decrease the stocking density, so that there is less stress and less mortality. Early morning or late afternoon transport is recommended, usually because of the conditions that prevail in that area, the climatic conditions and all. And the temperature also, because temperature is a big concern in this case. 25 to 50 ppm formalin treatment, just a kind of prophylactic treatment upon arrival for disinfection. I could have told you about, in case of fish seeds, we often use, diluted solution of KMNO4. Here we are using formalin treatment for prophylaxis. Then acclimatization period of over 1 week. We usually do not care about acclimatization or acclimation period, but that is one of the most important thing that makes the individuals adapted to the conditions where they are going to be kept. Stocking density, that may be 2 to 7 per square meter area, biomass not exceeding 300 to 400 gram per square meter area. Sex ratio, we can have 1:1, but sometimes higher female ratio may also be used, that to make it economical, to entire culture practice economical. how induced maturation is there? In fact, in case of male penates, there is no need usually to induce them for maturation. They generally mature in captivity, male prawns, but not the females. So, what we do for inducing maturity in captivity, um, either by eye stalk ablation, that is the endocrine part, or through nutrition or environmental manipulation. So, by manipulating different factors, whether physiological or nutritional or environmental, you can induce maturity in case of these prawns. Maturation in Penaeus monodon has been induced only through eye stalk ablation, although diet and environmental parameters may enhance reproductive performance. But how people have used and where from this content has been taken, what they have used, that is written, but all these manipulations can work, or you can take a combination of all these manipulations so as to get a better response. So, eye stalk ablation is very popular and that has been widely used for inducing maturation in case of prawns. How it is done? Females selected should be for eye stalk ablation. If you are taking, then they should be 80 to 90 grams, age 8 to 10 months. For pond stock, it is written healthy and hard shell. So, these are the criteria. In fact, brooders should be very healthy and of a certain desired range of size. Then thalicum should be uninfected, slightly bulging or convex, whitish vertical streaks. So, if you are having all these characteristics, then you can go for eye stalk ablation of that prawn. In crustaceans, decapod crustaceans, ablation of the eye stalk destroys the X organ and sinus gland that produces and stores respectively the gonad inhibiting hormone. So, by eye stalk ablation, what we are able to do? We are able to destroy the X organ and sinus gland. What they do? They, X organ produces this gonad inhibiting hormone, and this sinus gland that stores gonad inhibiting hormone. So, gonadal maturation inhibition is removed by removing the eye stalk or ablating the eye stalk. That is the basic concept behind this. So, the eye stalk can be ablated or destroyed by, there are many methods: incision pinching, cutting, or maybe eye can be removed totally or partially, some portion is removed. But this is very important point that appeals to me, that there are reports that a single eye stalk ablation is sufficient for inducing maturation in case of Penaeus monodon. So, why go for ablating both the eye stalks? Go for ablating only one eye stalk, and if one of them is already having some kind of problem, diseased or damaged, then go for ablating that eye stalk. Do not go for the healthy one, and by ablating even one eye stalk, you will get maturation quite good. So, this is how people are doing eye stalk ablation. There are many methods you can do that. Ablation during the post-molt leads to, these are some of the conditions that we should take care of. Ablation during the post-molt leads to mortality because of added stress. After molting, they are already in stress of getting infection or some kind of damage from outside environment. So, if you are doing eye stalk ablation during that time, that will be again very stressful to the animal, and excessive loss of hemolymph may occur. So, that is also one of the problems. Ideally, ablation should be undertaken during the intermolt for longer latency period of 2 to 4 weeks before maturation. So, if you follow these steps properly, you are going to get very good results if you follow the conditions what are recommended. The maximum number of spawns per molt cycle is greater for ablated compared to unablated Penaeus monodon. So, that way if we are applying this procedure, then we are getting good results. Wild subadult Penaeus monodon caught in mangroves took 40 days to mature and 69 days to spawn after ablation. So, Penaeus monodon was caught from wild. It took 40 days to mature and 69 days to spawn when ablation was done, compared to a minimum of only 3 days for wild adults from offshore, right. So, what you got from inshore and what you got from offshore, how the results are comparable? From the offshore, it took only 3 days, for them to spawn, whereas in case of uh, those which are caught from mangroves, they took longer time. Latency period may also be affected by the salinity. When wild Penaeus monodon from offshore Indian Ocean took only 4 to 5 days to spawn after ablation, in contrast to 20 to 30 days for females from brackish water. So, here it is more clear. In the upper point it was not very clear, but here in this last point you can see that Penaeus monodon collected from offshore took only 4 to 5 days to spawn, whereas after ablation of course, but whereas those which you collected from inshore in brackish waters, they took 20 to 30 days compared to 4 to 5 days. 20 to 30 days was the duration that was needed for the inshore prawns. Prerequisite to mating or spermatophore transfer is the molting of the female. So, that is basically one of the important steps. One male positions itself directly below the female, and by doing many such kind of activities, like finally curves and his body in a U shape around the female, flicking head and tail simultaneously, and ultimately, this is important, inserts the spermatophores inside the thelycum at this point of time. So, ultimately during mating, the spermatophores are inserted into thelycum. That is the important part to remember. Spawning. A ready to spawn female becomes restless and that's how they behave. Actively swims upwards in circles with the last 3 pairs of periopods held tightly together in flapping movement. Eggs are released through the gonopores simultaneous with the sperm release from the thelycum part over a period of 2 to 7 minutes. That is how spawning occurs. And once spawning is over, then eggs are generally kept in the same tank for fertilization until hatching. Nuclei are then collected and cleaned for any kind of additional tissue or something debris which is there. They are cleaned for transfer to larval rearing tanks and for transportation to other remote hatcheries, whatever your objective is. Time of hatching is 12 to 15 hours after spawning. Salinity preference or recommended salinity is 28 to 35 ppt. Temperature 26 to 29 degrees centigrade. Spawning tank should be chlorinated. Chlorination here in case of prawn culture, prawn hatcheries, that is one of the very important steps. Gentle aeration should be done. Maximum egg density that we should keep, we should not increase egg density from this, that is 3000 per liter of water. And the density, the water quality, these things should be monitored very properly. They are basically the source of the problems. If you are monitoring these things properly and feed, then everything will be fine. After spawning, the eggs are cleaned by removing any scum and passing the eggs through a series of meshes to remove both coarse and fine dirt particles. The main aim is to get them rid of any kind of associated undesired structures. Then stocking of nuclei. Nuclei are usually stocked in at 1 lakh per ton capacity of the tank and cultured up to late meiosis or early PL post larvae stages, with approximately 70 to 80% survival rate is generally expected. They are then transferred to a new tank and cultured up to post larvae stage, PL 12 to 15. A further 70 to 80% survival rate you may expect at this stage. Then diatoms, either Chaetoceras, Skeletonema or others that have been reared in monoculture for feeding them, are fed to approximately at 30,000 to 50,000 cells per ml. What that should be the concentration of these natural food per ml, starting from the protozoa stage to and continuing until early PL 4 to 5. So, at this initial stages, you should not go for artificial feeding. Try to give them these natural food items so they respond better. Artemia nopli, the probably one of the most popular food item of at this stage, at an average of 50 gram of cyst per 1 lakh larvae are given from meiosis to PL stage. So, and these are very popular, they give excellent results. From PL 4 to PL 15, artificial diets are commonly used to reduce the deterioration in water quality. As soon as you can go for artificial diet, then controlling everything becomes very easy. Otherwise, for preparing the enough number, enough quantity of natural food and then supplying it, and then after giving the natural food into your system, cleaning the system again becomes a challenge. So, all this you can avoid if you are able to feed artificial diets. So, from hatching it takes about 26 days to reach PL 15. Nursing of post larvae, the process of rearing them in nursery system after they are produced in a hatchery. PL 4, PL 5 are usually reared for 2 to 4 weeks in the nursery system before final stocking in grow out ponds. Here nursery systems supply the transition areas where young post larvae are acclimated to environmental conditions similar to grow out, and these nursing systems, what kind of nursing systems you can use? Post larvae can be nursed in concrete tanks, earthen ponds or cages, whatever kind of facility you are having, you can nurse them in those systems. A very brief overview of what are these systems is, in case of tanks, what we can do, the stocking density can be kept as 5,000 to 10,000 per ton of the capacity of the tank. Feeding phytoplankton and artemia, 5-day-old post larvae. After that, fed with muscle meat, trash fish and ascites. At again, these things depend on the availability of type of food at your place and the way you get more successful result. Water exchange 50% may be done. Siphoning of excess food, that is very important. In case of pond nursery, pond preparation that we have discussed at many places elsewhere in my lecture, this lecture or previous lectures. Pond area, usually 500 to 2000 square meter area is used for nursing. Stocking density 100 to 150 per square meter of area. And feeding with phytoplankton or artemia, I mean usually we prefer to provide natural food at these stages. Water exchange, maybe tidal water you can use for water exchange, 35 to 60 days culture period. Cage nursery, you can do these things in cages, and the cages we have talked earlier also that how they are made. And the stocking density here you can have 5,000 to 10,000 PL per cubic meter of water volume. Then you can, feeding is almost similar everywhere, that you can use, preferably you should use the natural food items, not artificial diet. Daily check the damage of the net and physicochemical parameters that we have already talked about. 35 to 60 days is the culture period. Now, how do we manage this system? Overall management practices that are involved in Penaeus monodon culture. Good quality formulated feed should be provided, that we have already talked in detail in my previous lectures. Avoid feed wastage. In the nutritional and feeding strategies, I have talked details of all these, so I am not repeating that again. Feed reduction during cloudy or rainy weather as well as during high temperature, or here I should add that even if you feel that the prawns are under stress or showing some kinds of disease or abnormal behavior, swimming or whatever. So, at that point of time, the feed should be reduced, or maybe if needed you can stop um, for that particular point of time. Weekly determination of growth, survival and FCR, that helps you to have a better account of how much you have fed and how much they need, whether there are more leftovers, so you can rework on what is the requirement, because feed is indeed one of the costliest input, in fact the costliest input in your culture practice. Water management, that we have talked a lot about this, that since we are culturing aquatic organism, so we need to take care of water like anything. That is the most important part. Regular monitoring of water quality parameters, especially some of the parameters like the temperature, the dissolved oxygen, the pH, and ammonia and other things that you need to take care of very well. So regular check of algal blooms or other things, if you have such kind of problems, removal of organic waste and extra feed, that should be taken care of properly and regularly. Disease management, if physical and behavioral examination of prawns, you should continue, as I have explained you in case of fishes also. Management practice, there are many common management concepts, principles that you can apply here, of course in a different system with a different organism, so you can do that accordingly. Physical and behavioral examination of prawns, that should be done on regular basis and continuously it should be done. Regularly check bacterial loads, in fact these things should be done on daily basis if possible. Regular check of feed consumption, use of good probiotics, sanitizer on regular basis. So these are some of the important management parts that you can have, and that takes care of I think almost all the aspects of Penaeus monodon culture. I have tried to cover almost every aspect of Penaeus monodon culture in all the available systems. Hope you enjoyed this lecture. Thank you so much.